Hi, good morning, everybody. I'm a PhD student in Florida International University Bioinformatics Research Group, led by Dr. Giri Narasimhan. My work is related to classification and ranking of protein docking models. So what is molecular docking? Given two proteins, A and B, docking aims to predict their best binding conformation. And it's very useful in many applications. For example, in vaccine design, we would like to know where antibody would bind an antigen. And the way how docking works is that it first generates many binding proposals. Those are binding configurations that satisfy certain criteria. And then as a second step, we need to score each of those and output the best conformation. But the problem with docking is that it generates too many candidates and it's very hard to rank. So the correct conformation is usually somewhere in top 100, but very rare in top 1. Therefore, the usual practice is to select top N models, let's say top 100, and then use it for further downstream tasks, such as parallel clinical trials. Therefore, if we improve the scoring of docking models, then it would save us tremendous amount of resources in applications such as vaccine design and drug design. The most popular scoring approach of docking models are empirical-based scoring functions. We aim to compute the binding energies of protein complexes and then combine it with empirical energy terms. And because the exact energies are too slow to compute, we need to employ approximations. And approximations usually cost with the cost of accuracy. Therefore, our goal is to use deep learning to improve the accuracy, but at the same time, maintain the efficiency of the runtime. You probably heard about AlphaFold. It's a turning point in computational structure prediction. They use deep learning to predict structure from its sequence, and they do it with pretty high accuracy. And motivated by the success of AlphaFold, we want to use deep learning and apply it for a problem of scoring protein complexes. Machine learning is a relevant approach here because there are thousands of protein complexes deposited to protein data bank. Those are experimental protein binding structures with detailed atomic information. Therefore, machine learning should be able to learn those binding patterns and generalize it for unknown protein pairs. So this is the list of examples of existing machine learning scoring functions for protein docking models. They are three-dimensional convolutional neural networks, graph neural networks, and geometric CNNs. I'm not going to go over all of those, only ones that related to my work. So one of the recent tools is called MASSIVE, which stands for Molecular Surface Fingerprinting, developed by Gainza et al. In 2020. So the idea is that we transform protein complexes into surfaces, and then we extract circular region on those surfaces at the area where the binding is expected to happen. So the data structure and input to the model is called patch. So the way how to describe it is to imagine a circular piece of wet cloth that you put on top of the protein surface, and the area that is covered by it is used to extract useful features. Those features are shape index, curvature, electrostatics, charge, and hydropathy. So then this circular piece is divided by grid cells or angular beams, and then each of those grid cells is associated with a feature. So then those patches are used as input to geometric convolutional neural networks, and it was trained in CME settings. There is also an improved version of MASSIVE called DMASSIVE. They skip data pre-computation part and they compute the molecular surfaces on the fly. Also, they improve the filters for geometric convolutional neural networks, and they report improved performance and runtime. So this DMASSIVE approach wasn't part of my original recon paper because I only recently discovered of its existence, but I executed it for datasets and added to this talk. This is the performance teaser for our tool. So we developed a deep learning approach called PIsToN that stands for protein interface, evaluating protein interfaces with transformer networks. So here I'm showing area under the curve and precision recall curves. So our line is red. So you see that we are above all the competitors, including massive and D-massive. So now, next I will explain how we achieved those results. So the first improvement is feature engineering. We started from the similar approach of patch extraction, and we added more features. So one is relative surface accessibility, and the other is distance between surfaces, meaning that for each point on the patch, we compute the distance to the complementary patch. And then in addition, we computed empirical energy terms. Those are fast approximated versions of energies. We don't have detailed information on surfaces. Those are just a single value for the complex. And one of our main contributions is that we project those patches into actual images. So instead of geometric objects with angular beams, now we have real images. So here, each pixel corresponds to a point on a surface, and the pixel intensities are values of those features. So we did it by projecting it through multidimensional scaling, and it's a pretty simple change, but it's very significant because now we can apply best practices from vision AI techniques. As a classification method, we used vision transformer. So we start from a protein complex from the training, and then we separate the chains, extract patches, and then we compute features, transform them into images, and now each feature is used as a channel. So then we divide the images into smaller pieces, and then each of those little squares is transformed into a vector, and then it goes as input to a vision transformer encoder. And the output of the model is embedding that's going to have a property of maximum separability that I'm going to describe later. And the important byproduct of this model is that it outputs an attention map, which we call spatial attention. It tells you the importance of each pixel on the input image. Therefore, it's endued with explainability, which is very useful for our application. The next improvement is that we enhanced the vision transformer with the hybrid component. I earlier mentioned that we extract approximated energy terms. We don't have the detailed atomic information on those. Therefore, we use a separate fully connected layer to get the representation of those energies. And then we concatenate the embeddings from VAT and then use another layer to get the final latent vector. So now it is aware of both detailed surfaces and also energies. So the final model is called multi-attention. So what we do is we divide the features into groups. So what we noticed is that pixels on different features has unique distributions. For example, you can see that shape index looks completely different from charge. Therefore, it may not be fair to use the same weights for all of the features. So we separate them by branches, and for each branch, we use independent hybrid network. So then we combine the representation from each branch using transformer encoder to get the final representation. And now, actually, we have even better explainability. From each branch, we get the spatial attention map that tells you the important pixels or important binding sites. And from the transformer encoder, we have feature attention that tells you the important group of features. So the next improvement is how we train it. So our training is we start from the native examples from protein data bank, crystallized structures. Then we separate the chains, and we use docking to generate docking models. So it generates as arbitrary number of structures. We take top 100, and then we label each of those examples using something called Capri criteria. So the idea is like that: we take each model and then we compare it with the original structure. If it's similar to the one from the experiment, we call it acceptable. If the binding site is somewhere far away, we call it incorrect. Then we construct a mini batch. We take arbitrary number of acceptable, 5 times more incorrect, pass it through the PIsToN network and generate embeddings. So now our job is to use contrastive learning. So what we do, we minimize the distance in the latent space of the same class and maximize this distance for different classes. At the same time, we introduce cluster centroids. Here it's shown as P plus and P minus, and we also separate those centroids in the embedding space. And the final score is defined as the difference in distances to the positive cluster versus negative cluster. To implement this contrastive learning, we combined 3 loss functions: binary cross entropy, supervised contrastive loss, and margin ranking loss. So then we performed extensive experiments on 2 well-known data sets. First is from original paper Massive, and the second is called Capri score, that's generated from the famous challenge to evaluate protein docking models. This is the final result. It includes state-of-the-art tools to evaluate protein complexes of docking models. It includes deep learning-based and empirical-based methods. Please note that it has more tools than in my original paper because we were informed about more competitive methods. For example, GNN DOF and DeepRank GNN DMassive. So what we see is that we outperform all of those tools in terms of area under the curve as well as F1 score, and we are competitive or better in terms of average precision, balance, accuracy, precision, and recall. And we consider AUC and F1 score as our primary metrics because it considers the balance between precision and recall. And also AUC contains all of the classification thresholds. Earlier I mentioned that our model is endowed with explainability. Here I'm showing an example. So we picked arbitrary protein. The PDB ID is listed here. So we run it through the network, and our network classified it as positive, which is correct because it's from the database. Then our feature attention indicated that hydropathy is a significant feature. And then we went ahead and visualized the interface map of the surface and applied the attention mask of the spatial attention. And it's highlighted this small blue region of where exactly the model paid attention. And then we mapped it back to the original structure. So you see those red residues. Those are corresponded to this significant area. And those residues were isoleucine, phenylalanine, leucine, and alanine. So all of those residues are hydrophobic, and it's at the center of interaction. Therefore, it's probably contributing to the hydrophobic interactions by minimizing the contact with water and making the binding more stable. So our model was able to highlight this effect. So in conclusions, I presented the deep learning approach for scoring protein docking models. First, we introduced the improved data representation by converting the protein surfaces into images that allowed us to use state-of-the-art image classification tools. In our case, it's Vision Transformer, and we also enhanced it first with hybrid component to accept empirical energy terms. Then we expanded to multi-attention that has dual axis of explainability. It highlights binding sites and important features. And finally, we introduced the way to train the based on docking models. We used contrastive learning to separate the embeddings, separate between positive and negative docking models. And all of this led to superior performance, which is going to be very useful in all of the docking applications, including vaccine design and drug design. So I would like to thank my funding sources that allowed me to travel to these amazing conferences. Those are from Florida International University, Graduate Assistance in Areas of National Need, then FIU GPSC and FIU Academy for CS Education. And the most I want to thank my advisor, Dr. Giri Narasimhan, who is leading Bioinformatics Research Group, and also all members of Bioinformatics Research Group for valuable suggestions and collaborations. Thank you so much. I'm ready for the questions.